The European Medicines Agency, or EMA, has qualified the PROCOVA methodology. Prognostic Covariate Adjustment, or PROCOVA, can increase confidence and potentially use smaller sample sizes for Phase 2 and 3 randomized controlled trials for regulatory decision-making. Is this a game changer for medicinal developers? Let's answer some common questions about PROCOVA. The first question is, what is PROCOVA? PROCOVA is a statistical methodology intended to improve the efficiency of Phase 2 and 3 clinical trials. It does this by using subjects' predicted outcomes on placebo to reduce the variance of treatment response estimates. The second question is, how does PROCOVA work? PROCOVA works in 3 steps. Step 1 is to train and validate a prognostic model. Step 2 is design. This involves using the model to plan the required sample size for the trial. Step 3 is analyze. This is where you use the prognostic scores for the final treatment effect estimation. The next question is, can I use PROCOVA for any of my Phase 2 and Phase 3 clinical trials? To use PROCOVA, you first need sufficient historical data and independent validation datasets. Secondly, you need a stable therapeutic landscape so that historical predictions remain valid. Lastly, the primary outcome must be continuous. This methodology is not for binary or time-to-event outcomes. The fourth question is, do I need to show the model dossier to the EMA? No, you do not need to show the model dossier to the EMA. However, you must validate the model and justify its use in the clinical trial protocol and statistical analysis plan. Next, why did the EMA qualify this methodology so readily for regulatory decision-making? PROCOVA strictly controls the type 1 error rate, which means the risk of a false positive. Methods like external control arm or Bayesian historical borrowing cannot. PROCOVA is a special case of the well-accepted ANCOVA method. It is mathematically proven to produce unbiased treatment estimates while keeping the type 1 error rate strictly controlled. The next question is, if it's basically just ANCOVA, why not just stick to the standard version? What's the actual value add here? PROCOVA can handle high-dimensional data, which can overwhelm standard ANCOVA. Question 7 is, what are the scenarios where this model just won't work? One deal breaker is low predictive power. Model performance, measured by the Pearson correlation coefficient R, needs to be high enough. Another is the safety buffer crunch. When applying safety buffers, you may not be able to reduce the sample size enough to maintain power. Finally, the linear limit. If the relationship between baseline characteristics and the outcome is a simple linear combination, PROCOVA does not provide an extra benefit. Given the conditions and uncertainty, a developer might ask about the return on investment, or ROI. It is one of the very few robust tools qualified for regulatory decision-making. Let's look at a use case in Alzheimer's disease, or AD, to illustrate the value of PROCOVA. In a reanalysis of the Quinn et al. Phase 3 study, using the prognostic score reduced the required sample size from 402 to 343 subjects. Cutting the enrollment by 15% is a massive win for the timelines and ROI of a Phase 3 study. Now that we have the full picture, when should I consider PROCOVA? Consider PROCOVA when your therapeutic area is relatively stable. Also consider it if you have access to a large volume of high-quality historical data, especially if baseline data is high-dimensional and complex. Finally, PROCOVA should be considered when the trial has continuous outcomes. Remember, it is not for binary or time-to-event outcomes. Please note that original insights, question framing, and content verification are done by a human expert, while AI assists with linguistic polishing, visuals, and formatting. This video is for informational purposes and is not medical or legal advice. For more information, please refer to the EMA qualification opinion for Prognostic Covariate Adjustment, or PROCOVA, at the link provided.